Good day, grade 12 learners, and welcome to this video where we will be focusing on RNA, the location, structure, and functions of RNA. In the previous video, based on the exam guidelines, we touched on DNA replication. We looked at the process of DNA replication. We also looked at DNA profiling, where we looked at the interpretation of DNA profiles, and we also looked at the uses of DNA profiles. In today's video, based on the exam guidelines, we will look at RNA in terms of location, structure, and function. We will look at the location of RNA, mRNA found in the nucleus and functions of the ribosome, tRNA located in the cytoplasm. We will look at the structure of RNA, it being a single-stranded molecule consisting of nucleotides. We will look at three components of an RNA nucleotide: the nitrogenous bases, a sugar portion, and the phosphate portion. We will also look at the stick diagram of RNA and tRNA molecules to illustrate their structure. And then lastly, we will look at the function of RNA, which is that RNA plays an essential role in protein synthesis. If we're looking at the location of RNA, there are going to be different locations of RNA. And remember that RNA is the second type of nucleic acid that we are looking at. RNA stands for ribonucleic acid. Now there are going to be different types of RNA, namely mRNA, which stands for your messenger RNA, as well as tRNA, which stands for your transfer RNA. Now mRNA, which is the messenger RNA, is located inside the nucleus, but it functions at the ribosome. So mRNA is located in the nucleus, but it performs its function at the ribosome. Secondly, we have tRNA, which is a transfer RNA. That RNA is located inside the cytoplasm, and it's generally also going to perform its function at the ribosome. So we have two types of RNA, mRNA, tRNA. mRNA located in the nucleus, functioning at the ribosome. tRNA located in the cytoplasm, functioning at the ribosome as well. If we have a look at the structure of a cell that we have here, we have the nucleus on the inside. So this purple section on the inside is the nucleus. We have our DNA strand located inside the nucleus. And then we have our mRNA strand also located inside the nucleus, but then it functions at the ribosome. Then you're also going to have your tRNA. Your tRNA will also come and they will also perform a function at the ribosome. Later on, when we look at the process of protein synthesis, then this will all make sense to you. If we look at the structure of our RNA molecule, it's generally going to be a single-stranded molecule, and that makes it different from a DNA molecule, because a DNA molecule was double-stranded. So an RNA molecule is single-stranded. It's also got ribose sugar, which is another difference compared to DNA, because DNA had deoxyribose sugar. Your RNA strand is going to comprise of 4 nitrogenous bases, namely adenine, guanine, cytosine, and uracil. Uracil is going to replace thymine, which we initially had in our DNA molecule. So in the DNA molecule, we would have had bases adenine, guanine, cytosine, and thymine. However, in RNA, that now changes to adenine, guanine, cytosine, and uracil. If we have a look at the diagrammatic representation of an RNA molecule, you will notice firstly that it's a single-stranded molecule. You only have one single strand here. Once again, you are going to have your nucleotides as the building block of the RNA molecule. The type of sugar that is evident here is ribose sugar. It's not deoxyribose as in the DNA molecule. And the possible nitrogenous bases will either be adenine, guanine, uracil, or cytosine. So that is the basic characteristics of the structure of a RNA strand. In addition to that, you will need to be able to tabulate the differences between the DNA and RNA molecule in terms of structure and in terms of the nucleotides. So if we have a look at this table over here, it simply shows you the differences that exist between DNA and RNA in terms of structure. This is a question that is examinable, so please make sure you are able to provide at least 3 differences between DNA and RNA in terms of structure. So if we have a look at the 3 differences, firstly, DNA is a double-stranded molecule, RNA is a single-stranded molecule. DNA contains deoxyribose sugar, but RNA contains ribose sugar. DNA contains nitrogenous base thymine, but RNA contains nitrogenous base uracil. And these are the 3 distinguishing differences between DNA and RNA in terms of structure. Now, if you are asked to differentiate between DNA and RNA in terms of the nucleotides only, then you are only going to focus on the last 2 differences. Because remember, the nucleotide comprises of the phosphate group, the sugar, and the nitrogenous base. So you can only state differences based on those characteristics if asked to differentiate between DNA and RNA based on the nucleotides. You will then say DNA contains deoxyribose sugar, RNA contains ribose sugar. DNA contains thymine and RNA contains nitrogenous base uracil. Just one last thing to note, please ensure that when you are asked to differentiate between DNA and RNA, and if you are asked to tabulate the differences, please ensure that you are drawing the table, as a mark is always awarded for actually constructing that table in an exam. Now, if we are looking at the function of RNA, RNA has one main function, and that is that it plays a role in the process of protein synthesis. So RNA plays a role in the process of protein synthesis. So in summary of this video, we looked at RNA in terms of location, structure and function. We looked at the location of RNA, the structure of RNA, 3 components of the nucleotides. We see that they are nitrogenous bases, adenine, uracil, cytosine and guanine. It had a sugar portion, namely ribose sugar. It had a phosphate portion. We looked at the stick diagram of mRNA and tRNA to show the structure. And then lastly, we see the function of RNA is that it plays a role in protein synthesis. In the next video, we will focus on the process of protein synthesis.